phospholipid is a lipid made of glycerol, two fatty acid tails, and a phosphate-linked head group. Biological membranes usually involve two layers of phospholipids with their tails pointing inward, an arrangement called a phospholipid bilayer. Phospholipids arranged in a bilayer make up the basic fabric of the plasma membrane. They are well suited for this role because they are amphipathic, meaning that they have both hydrophilic and hydrophobic regions. As shown in the figure, each phospholipid molecule has a head and two tails. The hydrophilic, or the water-loving portion of a phospholipid, is its head, which contains a negatively charged phosphate group as well as an additional small group, which may also be charged or polar. The hydrophilic heads of phospholipids in a membrane bilayer face outward, contacting the aqueous fluid both inside and outside the cell. Since water is a polar molecule, it readily forms electrostatic interactions with the phospholipid heads. The hydrophobic, or the water-free part of a phospholipid consists of its long, non-polar fatty acid tails. The fatty acid tails can easily interact with other non-polar molecules, but they interact poorly with water. Because of this, it is more energetically favorable for the phospholipids to tuck their fatty acid tails away in the interior of the membrane, where they are shielded from the surrounding water. The phospholipid bilayer formed by these interactions makes a good barrier between the interior and exterior of the cell because the water and other polar or charged substances cannot easily cross the hydrophobic core of the membrane. Now that we have discussed the basic fabric of the plasma membrane, let us now then go to the components of a phospholipid bilayer.
Proteins are the second major component of plasma membranes. There are two main categories of membrane proteins, integral and peripheral. Integral membrane proteins are permanently embedded within the plasma membrane. They have a range of important functions. Such functions include channeling or transporting molecules across the membrane. Other integral proteins act as cell receptors. Peripheral membrane protein. Peripheral membrane proteins are proteins that are only temporarily associated with the membrane. They can be easily removed, which allows them to be involved in cell signaling. Peripheral proteins can also be attached to integral membrane proteins, or they can stick into a small portion of the lipid bilayer by themselves. Glycoprotein and glycolipid. In the cell membrane, carbohydrate chains can be covalently linked to membrane proteins to form glycoproteins. They can also be covalently linked to membrane phospholipids to form glycolipids. Glycoprotein and glycolipid form hydrogen bonds with the water molecules surrounding the cell and thus help to stabilize membrane structure.
Lastly, we have the two components of a phospholipid bilayer, which are the protein channel and the cholesterol. A channel protein is a special arrangement of amino acids which embeds in the cell membrane, providing a hydrophilic passageway for water and small polar ions. Like all transport proteins, each channel protein has a size and shape which excludes all but the most specific molecules. The cholesterol molecules are randomly distributed across the phospholipid bilayer, helping the bilayer stay fluid in different environmental conditions. The cholesterol holds the phospholipids together so they don't separate too far, letting unwanted substances in or compact too tightly, restrict free movement across the membrane. Without cholesterol, the phospholipids in our cells will start to get closer together when exposed to cold, making it more difficult for small molecules like gases to squeeze in between the phospholipids like they normally do. Without cholesterol, the phospholipids start to separate from each other, leaving large gaps.